Okay, so today will be the last, or the second conversation really on the urinary system. The first discussion was last Tuesday. You've been provided two, one was about 45 minutes on the male reproductive, one was about an hour on the female reproductive system. I know that your lab practical is your primary focus right now. And once you get past the lab practical, I suspect that you'll go in and listen to those lectures if you haven't already done so. You'll also notice that I did spread out the homework just a little bit more than I did last time. So fair warning to all those who are listening in Cyberlander who are here right now. I did stretch out the homework assignments because last time too many people were waiting to the last second to do their homework. And it just wasn't as effective. It's not useful to do the homework after the exam. It doesn't make any sense. So I did spread those out just a little bit more so that you'll get them done sequentially.
I just want to tell you thank you for that because it makes it easier in my life.
Well, I mean, you don't have to wait to the deadline, right?
No,
but there's a mentality, right? There's a mentality to let me wait, let me procrastinate until the very, very end. And then it's like, uh-oh, I've got all this to do, and I really can't learn it all that fast. So I think it's better. I hear complaints no matter what I do. I'm used to it, right? No matter what I do, someone's not happy. It's a benevolent dictatorship. What's that?
Just like at home.
Just like at home. You know my family, do you? You've been talking to my wife. Oh, no. So absolutely, right? No matter what we do, no matter what our intentions are, someone's not happy. But in the end, hopefully we're doing it for the right reasons. So 3 topics on the next final, on the last final exam, not a cumulative final, simply a discussion over reproductive, male and female, urinary, and some acid base that I'll be discussing for you, which comes from Chapter 25. The last part of Chapter 25 we'll be finishing up on Thursday. Recall that we started on Chapter 25. We started our very first day talking about water balance and sodium and potassium and where they're found extra and intracellularly, and then that conversation from sodium and potassium led us into action potentials, and off we went with the nervous system. Now we come back to Chapter 25, starting Thursday, to discuss acid-base regulation and how the kidneys are involved with that, and also how the respiratory system is involved with maintaining our acid-base levels. So I think it kind of comes around in a nice way. Any thoughts or questions right now related to the urinary conversation from last time? Let's think about talk to me about the kidney. What do we know? Just popcorn out some ideas. Let's do anatomy first. This is good for your review for the exam anyway. So just talk to me about the kidney. Pieces and parts. If I cut into the kidney, what do I see? I see a cortex on the outside. I see a medulla. On the inside of the medulla is made up of pyramids, 8 to 15 of those pyramids. If I go microscopically down into the cortex and into the medulla, what would I see under the microscope? Well, microscopically within the cortex and within the medulla, I'm going to see tubes, right? Nephrons, right? Tubes. And most of those tubes are lined by what kind of epithelium? Simple cuboidal, right? Most of the proximal and distal convoluted tubules, the loop of Henle, most of it's simple cuboidal. We've known that since lab 3 of 105. What else am I going to find? All the renal corpuscles. Where are all of the renal corpuscles? Cortex or medulla, or mixed? All of the renal corpuscles, all the glomeruli with the Bowman's capsules are where? Up in the cortex, right? So in the cortex is the only place you would find any of the renal corpuscles, which is, of course, the Bowman's capsule surrounded by the glomerulus surrounded by the Bowman's capsule. What's going into every single one of these renal corpuscles? What's bringing blood in? Afferent arterial. What's bringing blood out? Efferent arterial. What process is happening in the glomerulus? Filtration under a certain amount of pressure. Then where's that blood going? As it leaves out the efferent arterial, it's going to head to one of 2 capillary beds, regular continuous capillaries, either the peritubular capillaries or the vasa recta. Remember this? Briefly, a little bit. Now, once the plasma is filtered, we've just crossed over the fenestrations and the filtration slits caused by the podocytes within the Bowman's capsule. That filtrate is now going to begin a journey down what? Am I hearing proximal convoluted tubule? Right? And what process is essentially happening in the PCT, in the proximal tubule? Reabsorption, right? Because I told you last time that about 150 liters a day of filtrate are being produced by women, about 180 liters a day are being produced by men. we've only got 3 liters of plasma. We've got to recapture most of this. Tommy?
I wasn't sure from the video, but is it both had peritubular? And
Both have peritubular, only the juxtamedullary nephrons with the deep, long loop of Henle have the vasa recta.
So they both go to peritubular first, and then one of them goes to
Yes, if it's a juxtamedullary, the peritubulars would also have a branch coming off the vasa recta. We'll get more to that today, so we'll get there. I promise you, we'll get there. So we got reabsorption happening in the proximal convoluted tubule. That's not to say that other things aren't happening there, but that's the primary job of the proximal tubule. Then the loop of Henle, what's happening? This is what we're going to pick up today. But as you dive down into the loop of Henle and come back up, it's sort of like diving into the deep end of a swimming pool. As you go deeper down in the deep end of the pool, what happens to the pressure? Pressure is increasing, isn't it, as you go deeper into a pool. And that's exactly what's going to be happening as we dive down that deep loop of Henle. There's going to be a greater osmotic pressure that's going to occur. And what that's going to do is to further concentrate the urine. It's going to drive more water out of the urine and concentrate those waste products within the tube. And then it comes up, and now we're at the other end of the distal tubules called the other end of the convoluted tubules called the distal convoluted tubules. And what did I tell you was a primary job here? Is that kind of where we dropped off? But what's the process is happening? It's happening everywhere, but largely in the DCT? Secretion. And secretion is the opposite of reabsorption. So what is reabsorption, first of all? The movement of molecules from the tube back into the blood capillaries. Secretion, therefore, must be taking things directly from the capillaries, from the blood, and putting them into what will become the urine. It's not urine yet, right? It's still a filtrate as we go through the nephron. It's not until we're completely through the nephron and all the way through the collecting duct has the urine reached its final composition. Okay. Is there anything I've just said that doesn't resonate with you, not making sense? So far, so good? Now, Tommy's question about what are these, today what we're going to be talking about is what's this vasa recta? What is this capillary bed that's going down along the loop of Henle? And what is its purpose? That's really where we're going to head primarily over the next little while. Is this about where I left off? I want to say 24:9, 24:10. I just described, there's 2 more things I want to review with you. Let me go back a couple of slides. I think we need to have a picture of this. Let me ask you, what is the significance of this area? What's happening there? What was coming back and talking directly with the afferent and efferent arterioles, bringing blood into and out of each and every glomerulus? Yeah, the distal convoluted tubules, right, is coming up. Actually, the loop of Henle is coming up. And it's going to become the distal convoluted tubule. If you want to think of a place where the loop of Henle becomes the distal convoluted tubule, think of it pretty much at this intersection. And this tube comes up and does what? Speaks to directly through hormonal and hormonal signals and stretch receptors and talks to the cells wrapped around the afferent arteriole. What were the cells right here in the middle called? Mesangial, mesangio in the middle of the vessels, right? The mesangial cells. And I don't think I said it last time, but there's a group of cells right here, that block of cells called the macula densa. We'll talk about that too. What does macula mean to you? Spot, macular degeneration, an eye deficiency where people start seeing an eye, a spot in their visual field that gets bigger and bigger. So macular means a spot. So what's macula densa mean? A dense spot, right? It is simply a group of cells. And those cells are sitting right there at the intersection of the mesangial cells, and they're sitting inside the distal convoluted tubule. So we'll come back to those. What do we call this whole complex? The juxtaglomerular apparatus, right? The JGA, juxtaglomerular apparatus or juxtaglomerular complex, okay? Complex or apparatus. You'll see both words used in the literature. And what was happening here? There's three ways that blood flow is controlled through a kidney. There's local regulation, there's endocrine regulation, and there's neuroregulation. How would you describe how these cells are interacting with each other?
Local.
Auto, local, neuro, or endo? There's no hormones here. Local. So auto-regulation or local regulation. And what did I tell you was happening here? If there was too much fluid in the distal convoluted tubule, then as that fluid's coming up, it's going to stretch some of these cells of the macula densa. And what are those macula densa cells going to say to the afferent arteriole? Squeeze. Right? And what would those cells of the afferent arteriole be doing when you squeezed? Restricting the flow of blood into that particular nephron. So that is an auto or a local control mechanism. So every single nephron is self-regulating the amount of fluid that is able to go into it. Now, a nephrologist, right, a kidney specialist, can't deal with looking at each and every individual nephron. There's millions of them. So from a pharmacological standpoint, from a physiological standpoint, a nephrologist is thinking about the entire kidney as being one giant nephron. Right? All they can do is treat the nephrons collectively, right? The millions of them pharmacologically together. But this is what's going on. This is a local regulation. What do we call those cells wrapped around the glomerulus again? Those cells that are wrapped around here are the what? Podo or podocytes, right? And those foot-like extensions that wrap around and form that filtration slit. There's also another term that I don't think I used last time to describe the overall orientation of the renal corpuscle. This is considered the vascular end. Why is this the vascular pole, actually? The vascular pole. This end is the vascular pole of the renal corpuscle. Why? Because there's blood going in and out of the corpuscle. So this is the vascular pole, and this side, what do you think? Like polarity, right? North and south pole. So if that's the vascular pole on the left, what would be the right side? Any idea?
Avascular?
Not avascular. It's a good guess, though. You'll see it called a couple different things. Vascular pole and also having one of those senior moments. I'll come back to it. How's that? I don't want to tell you the wrong word. I'll think of that word, and it goes right here. We'll fill it in, okay? And I want to say urinary pole, and I think that's right, but all of a sudden I'm wondering, I'm guessing myself. So we'll hold on to it. It's not a major description, but you'll hear about the renal corpuscle having, there's polarity, right? There's different sides to it. And so the afferent and the efferent arteriole always come in on the vascular pole, and the proximal convoluted tubule always goes out on the, we'll call it renal pole for now, okay? It'll come to me. So that's kind of where we were. Is there anything in that conversation that's not making sense?
The mesangial cells, is that right at the juxtamedullary?
It is in, see these little yellow cells in here? Oh, let me
Yeah,
turn this
that's
back on.
Okay.
Okay, those cells in this little area, in between the vessels. So they're in between the proximal tubule, or sorry, the distal tubule and these yellow cells. Those are the mesangial cells. Yep. We'll come back to those a little bit and talk about it. So this is autoregulation. Okay, this is self-regulation. It could also be considered, but that's not all that happens, right? We also have the ability of bringing in central regulation. That is, the body can now speak to the kidney through either endocrine signals or through nervous signals. Now, you already know the nervous signal. What's influencing the flow of blood into and out of the kidney? Parasympathetic, sympathetic, right? So the autonomic nervous system is already has an influence on the overall blood flow going to the kidney. That story we know. There's also some endocrine functions that are happening. The juxtaglomerular complex, those cells, can form a hormone called renin, or renin, R-E-N-I-N. Now, that makes the kidney an endocrine organ, doesn't it? Okay? Now, there's another hormone made by the kidney. Can you think of what it is? We talked about it previously. The kidney makes renin. I'm talking about it now. Not the angiotensin. The angiotensin actually is made by the liver and is flowed. We'll get to that story. But what about, no, aldosterone is made by the, what's the adrenal gland? Okay, close by. What's another hormone that's made by the kidney? Made by the hypothalamus, released by the posterior pituitary. How about EPO, erythropoietin? Remember erythropoietin, right? EPO, erythropoietin, and it's made by the kidneys, influences red blood cell production. So we've seen the kidney before as an endocrine organ. It makes the hormones. It certainly is responding to hormones as well. So now the hormone we're thinking about is renin. Now, you just mentioned angiotensinogen, so let me get into that story. Renin is going to travel through the bloodstream and convert a protein called angiotensin 1. Now, I know this is too small for you to read right now. Angiotensin 1. Angio, what's this protein do? Angio, vessel. Tensin makes them more tense. Okay, so just the hormone's name, or sorry, the molecule's name kind of tells you what it's doing. And what this is going to do is activate, angiotensin 1 is in the blood and is going to be activated to become angiotensin 2. So 1 becomes 2, it's just in order. And the molecule that is going to do this is ACE, angiotensin converting enzyme. Hold on. This is a strange little complex, but it's really, really important to understanding overall blood pressure. So when you're looking in your book, and I need you to look in your book, and I need you to do the homework that's going to help you with this. Angiotensin is activated by renin, right? So renin is basically a hormone enzyme. Renin tells angiotensin 1 to become angiotensin 2, okay? And then, sorry, I just misspoke. Renin is going to form angiotensin 1. Angiotensin 1 will then become angiotensin 2 through ACE. Now, this enzyme is largely made in the capillaries of your lung. It's kind of a weird story. There are specific cells in the lung that make this ACE enzyme. Now, you'll hear about people having ACE inhibitors, right? And those ACE inhibitor medications are actually inhibiting or interacting with this ACE enzyme. So this is happening in the peripheral blood, isn't it? I mean, we're releasing a hormone from the kidney. It's going through the blood. It's causing angiotensin to be formed. And then angiotensin 1 is being converted to angiotensin 2 by ACE in the lungs. Now, angiotensin 2 is a very, very powerful vasoconstrictor. It's causing tension on the angios, on the vessels. If you squeeze on the vessels, what's going to happen? Blood pressure is going to go up. So why would we give individuals ACE inhibitors? To block this angiotensin 2 from being formed, block this vasoconstriction process, and interfere with the ability to raise the blood pressure. Okay, so ACE inhibitors are going to be blood pressure reducing meds, and they interfere with this pathway. So in a normal physiology, though, you have this ACE, right? And what's that going to do? Why would the kidney be making renin? What do you think the trigger is for renin? I'm telling you that it's causing high blood pressure. So what must the trigger be for renin? Low blood pressure, right? So if the pressure coming to the kidney is not sufficient, then the kidney spits out renin. And what does renin do? With the help of ACE, makes angiotensin 2. Angiotensin 2 is going to tense on the vessels and increase the pressure. So this would be an endocrine connection, wouldn't it? An endocrine regulation loop. So now we're going to, not only is it going to constrict the peripheral arterioles. Look what else it does. It also further constricts the efferent arterioles. Think about it. If you're going to squeeze down on the peripheral arterioles, you're going to raise the blood pressure. If you're squeezing down on the efferent arterioles, leaving the glomerulus, what's happening to the amount of blood being filtered? You got to go on the magic school bus ride with me. You have to imagine you're down at the glomerulus, and you are a blood vessel, you're a blood cell coming into the glomerulus. And I've just closed down the efferent arteriole. What have I just done? I just shut down your exit, didn't I? Do you agree? The efferent arteriole is the way that blood leaves the glomerulus. So if I'm going to squeeze down on the efferent arteriole, what's going to happen to the amount of filtration happening? Tommy? Up, right? Because what are you doing? You're causing the blood to spend longer in the glomerulus. The blood is going in. You've already increased the pressure, right? Peripherally, you've got more blood going into the glomerulus under higher pressure, and you squozen down on the efferent arteriole, so now blood can't get out. So what's going to happen to your GFR? Your glomerular filtration rate. It's going to go up, right? You've just pushed more blood into the glomerulus, and you closed the back door. Didn't close it, kind of made it slower, right? Close down the back door a little bit. So what's that going to do? It's going to increase your blood pressure, right? And restore some of this flow. Because why in the world did the kidney release the renin in the first place? Because there wasn't enough blood pressure. Remember I told you, I think I alluded to this, if the kidneys are not getting sufficient oxygen and sufficient blood pressure, they get pissed off and they send off alerts. So they're sending out alerts about low oxygen in the form of erythropoietin. Let's make more red blood cells. Let's get more oxygen to the kidney. And they're sending out renin in response to low blood pressure, causing angiotensin II with ACE to increase that pressure and shut down the efferent arterioles exit. That increases the GFR, increases the filtration rate happening at each and every glomerulus. Okay, so there's one more thing. This is a complicated little pathway, and I need you to know this figure. Right? I know it's too small for you to memorize from this. You're going to have to crack your book and look at this. I'm probably going to have one, well, I'm announcing it now. I just made up my mind. I'm going to have one short answer question on the test, and it's going to be to tell me about this pathway. I need you to be able to tell me about renin and about the juxtaglomerular apparatus that's making the renin, and tell me what happens in response to low oxygen and low pressure, and how it is now going to affect angiotensin and use the ACE enzyme. So I need you to know this story. Now, the other thing that angiotensin II is doing, maybe you saw this other hormone up here, aldosterone. Angiotensin II is also traveling in the blood and heading to and affecting increased aldosterone. Now, what is aldosterone being released by? The adrenal glands. Remember the salt, sugar, and sex layers of the adrenal gland? So this is that salt regulation, isn't it? So this would be from the outer layer. Salt, sugar, sex. Remember that? Maybe? Okay. So the outer layer was the salt layer, and aldosterone is going to be released in response to angiotensin II. What does aldosterone increases sodium retention? Okay, so it's working on the kidneys. It's increasing sodium retention. In other words, what's it doing? Reabsorbing more sodium. We learned in Biology 105, Lecture 1, Lab 1, Day 1, pretty much. Solutes suck. If I'm reabsorbing more sodium in the kidney, what's following? Water. Increased water in the body is going to do what to blood pressure? Increasing it. Do you see we've got multiple ways that we're increasing blood pressure? We've got renin, we've got ACE, we've got the efferent arterioles, we've got angiotensin, and we've got aldosterone coming in. All of this is so cool, right? So with increased sodium retention, we're going to increase our fluid consumption. We're going to increase our blood vessels and blood volume. That's going to increase our blood pressure. It's also going to constrict your venous reservoirs. What's that going to do? Where is most of your blood right now sitting at rest? Arteries or veins? It's in the veins, right? About 60% of your blood is sitting in your venous side at any one moment. So it's also going to help constrict those venous reservoirs and push that blood into, right, your arteries, increase your blood pressure. It's also increase your cardiac output because it's also going to increase angiotensin II, also increases your sympathetic tone, right? Sympathetic tone referring to the vessels. So it's going to increase your cardiac output, again, increasing your blood pressure. It's a beautiful thing, but there's a fair amount here. So I want you to know this story. And basically, I will give you this question next time, and I'll post it. And basically what I'll do is I'm going to give you 10 or 12 terms. It takes me forever to grade these, but it's good medicine. I'll give you 10 or 12 terms, and you'll have to create either a flow chart or a concept map or a paragraph that links these words together. I'll let you choose. Is it more of a flow diagram or is it more of a paragraph with words? But you'll need to put these 10 or 12 terms, and I'll give you the list. And I'll give that to you before I see you next time. And that will be your one and only written assignment on the exam. Okay, you're going to know what it is in advance. There's no reason why you don't do really, really well on it. But I'll give that to you, and then you can talk to me about it on Thursday when I give it to you. And remember, we have a review session next Tuesday. So Thursday, I'll finish up with acid base. If there's any dangling concepts, we'll finish them up at that review session and also entertain any questions, and we can certainly even go to the darn board and walk through this if you want to do that as a group next Tuesday, and then our exam will be a week from Thursday. Fair enough? Is this making sense? Or are you all focused on the lab exam? I understand, right? There's only so much we can do. But it does make sense. It really, really does as you go back and lay this out on a whiteboard or on a piece of paper. Oops, okay. So, very important, slide 70. Slide 70, very, very important. And as you read through your book, it'll help you. Oh, come on. There we go. So that was 24.9. Okay. Now, let's go back into these processes that the kidney does. Reabsorption, secretion, the concentration of the urine. And I've already said this, so some of this should be review. So what is the primary job of the proximal convoluted tubule? Reabsorption, right? Now, is there some secretion going on there? Oh, yeah, there is. There's some secretion going on there, but its big job is going to be reabsorption. And 99% of the glucose, a lot of the water, the sodium, the potassium, the other electrolytes are all going to be reabsorbed. Now, as we reabsorb these things, where are they going, folks? What's wrapped around the proximal tubule? What capillary bed is wrapped around the proximal tubule? The peritubular capillaries, right? So as we reabsorb out of the tubes, where is it going? Into a capillary bed. And we know that capillaries are the place where things exchange. So this is where water and electrolytes and glucose and all the other molecules that are going to be moving in and out of our tissues are exchanging across that simple squamous epithelium of the capillaries. Now here it has 108 liters each day of water is going to be reabsorbed, right? Lots and lots, and you'll see different numbers. But I gave you a number of 150 to 180 liters a day of filtrate. So regardless, don't memorize the number, it's a lot, right? It's a lot of fluid that is going across the glomerulus. Now what's going to happen here? Water is going to move into the peritubular fluid, right, around, and the solute concentration of the tubular fluid is going to decrease as this water is coming in. So let's make sense of this. So we're in the proximal convoluted tubule. Do you agree that's where I am in this purple area? Notice there's arrows. The arrow, the blue arrow, right, and the orange arrow are going out of the tubules, out into the space around the tubes. What process is that? That's reabsorption, right? And what's going to reabsorb these water and electrolytes will be the capillaries that are floating around this. You also see on the bottom a little inward arrow. So I don't want you to think that secretion is not happening here. There is some movement, there is some secretion happening here, but it's more of the reabsorption story happening here. Again, the tubes are going to have what around them? What kind of epithelium? Simple, cuboidal, and we need to increase the surface area here. So just like in the small intestine, there would be lots of microvilli. Right, so lots of microvilli, increasing the surface area for this reabsorptive process. Look at this figure, the top, it looks like a sun. That's the tubular fluid. That's the lumen of the tube. And notice it's got all the little microvilli enfoldings on there. And the other thing this is trying to show you is that the reabsorptive processes here are specific. This is transcellular movement, meaning that the molecules going from the lumen back into, right, here's the peritubular capillary. Molecules that move across one side of a cell are transported through the cell and come out the other side. That does not happen randomly. This is specific, so there are specific transporters, processes that carry things through the cell. This is a directed process. What is going to be reabsorbed will be the water. Now, there's going to be some water that is going to come squiggling through the cells, but most of it is truly going to be welcomed right through by osmosis. Okay? And then the ions are also coming in. Glucose is coming in. Now, glucose can't just cross the membrane. There are transporters here. There are proteins found on the apical side, right, the luminal side of the cells, and glucose is brought into and through the cell. And then selectively, this would be do you agree this would be the apical side? My finger going here. Do you agree that this would be the apical side of the cells? And this we would call the basal side. And then down here is the peritubular capillary. So we have molecules moving through here. Okay. Now, here's our key. This blue arrow is water reabsorption, and the orangey arrow is solute reabsorption. Regardless, it's movement of molecules out of the tubule going into the blood. Okay, so that's what's happening in the proximal tubule. Let me go back to that. Look at this arrow. Again, I don't want you to think that secretion is not happening. This red arrow here, there is secretion going on. Okay, so there are going to be molecules coming from the blood or from the cells and being dropped directly into the tube. So secretion, right? The opposite of reabsorption, the movement of molecules from the blood directly into what will become the tubule. Only about 15% or so of the initial volume of fluid is going to make it to the distal convoluted tubule. Right? That means that we have reabsorbed a lot of water, haven't we? A lot of water is being reabsorbed in the proximal tubule and even a little bit down along the loop of Henle. As the processes of reabsorption and secretion are happening, what's happening to the filtrate? It's becoming less and less like plasma and more like urine. Right? That's what's happening through this whole process. It was plasma at the very beginning, wasn't it? And it's becoming more and more like urine as we add and take away from this fluid. Now, sodium is reabsorbed in exchange for potassium. So what does that mean? If sodium is being reabsorbed, potassium is being the opposite, secreted. So wherever you see sodium being reabsorbed, potassium is being spit out in exchange for that. And this exchange is being stimulated by aldosterone. What was aldosterone's story a moment ago on that chart? What did aldosterone do? Aldosterone increased the sodium reabsorption. It increased fluid levels and blood pressure, didn't it? So the opposite of that would be potassium is going to be secreted. So what's happening to potassium? It's going to end up where? In our urine, right? So potassium is definitely secreted. Hydrogen is also secreted. Talk to me about this story. How What's our evidence from lab last week that hydrogen is secreted? What's our blood pH? At or about 7.4, right, Roxana? What's the pH of our urine? You saw it in the chem strips. Acidic, right? Usually less than neutral. So what does that tell us? That in order for the body to maintain that perfect 7.4 blood pH, it must get rid of any extra acid. How does it do it? By putting that acid into the urine. By what process? Secretion. So we know that acid is being secreted, and I'm telling you that potassium is also secreted. Those are the two big ions that are being secreted, things taken from our blood and put directly into what will become our urine. Now, there's also other things being secreted and released into our urine. I mean, this is why when you go to a new job, they do a urine test, right? They're looking for metabolites. Other medications and drug use is picked up in the urine. So there are also other molecules that do end up in our urine and are also secreted. Some toxins, some drugs are also released or, quote, secreted into the urine through this process. So what does this look like? In a diagram like this, now, again, we're in the distal convoluted tubule. That's not to say that both processes are not happening here, but the primary job of the DCT is secretion. And what do we see now? Things are going from the bloodstream. Here we are. Right? We're down in the peritubular capillaries. We want to put this stuff now into the tube to get rid of it through the urine. So what things did I say are being secreted? What were my three things? Potassium, hydrogen, toxins, and drugs. All right, they're there. Those are those three molecules that are being taken from the blood and secreted directly. Notice again that this is a selective process. I mean, these molecules are being welcomed into the cell and are being put into the tube. Guess what that requires? Energy, right? ATP, right? There's energy involved here. There's a lot of energy spent. A lot of ATP, a lot of mitochondria are making energy, right? So that these processes can happen. Many of these are active transport. What is active transport? Remember this term from long ago? What's passive? Things going from higher concentration to lower concentration without the use of ATP. So what is active transport? When you need ATP, why? Because you're moving up the gradient. You're moving from an area of lower concentration and you're already putting it into a place where there's already stuff there. If we depended upon passive transport, would our urine be as acidic? Right? We're packing acid molecules into our urine. It would not go there naturally, right? Because it's already acidic. So we've got to push it there through this idea of active transport. It takes energy, ATP, mitochondria, pushing this process. So these little yellow guys, transport pumps. Pump suggests there's some energy behind it. So there's definitely some ATP that's pushing this. So what would there be a lot of in these cells? If they need a lot of energy, they need a lot of ATP, there would be a lot of what organelle? Mitochondria. So you better believe that there's going to be a lot of mitochondria in these cells of the tubules because they have to generate this energy to pump these molecules to where they need to go. What do you think? Is there reabsorption happening in here in the DCT? Oh yeah. But the big thing here is secretion. Do those two processes make sense? So name for me 4 things that are reabsorbed. Sodium, glucose, small proteins, magnesium, calcium, right? All of the electrolytes, chlorine, right, would all be reabsorbed. Name for me 3 things that are secreted. Potassium, hydrogen, and toxins. Now, this next couple slides is where we need to really focus in. This is, to me, a difficult concept because, and the reason it's a difficult concept is because the drawings that we see in textbooks oversimplify the nephron. When you look under the microscope and you're looking at a slice of kidney, what do you see? Tube, 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 right? Densely packed tubes. When you look at a cartoon, an artist's representation of the kidney, what do you see? A nephron over here, oh, and a nephron over there, and another nephron over there. But they're not tightly packed, are they? You need to imagine that these loops of Henle and these proximal convoluted tubules are not just isolated in their own little neighborhood. They are packed together and are touching each other. And what about those capillaries, those peritubular capillaries? Is one peritubular capillary dedicating its life to wrap around one set of tubes? No, right? It's this dense, packed-in environment. So there would be peritubular capillaries wrapped around all the tubes. There'd be vasa recta wrapped around all of the loops of Henle, but in a more densely packed way. I'm saying that because as I describe this, you can't rely upon the simple diagrams of the textbook. They're there to help you begin to visualize this, but it doesn't make sense unless you realize these things are all coexisting in tight locations. So this idea of countercurrent, the thin limb and the thick limb. Look at these terms, thin descending limb and the thick ascending limb. We're talking about loops of Henle, right? The nephron loops. They're located close to each other. They're separated by fluid, peritubular fluid, right? The fluid that's outside the tubes, that's outside the capillaries, would just be called the peritubular fluid. Where are we in the nephron? Or sorry, where are we in the kidney? Loops of Henle are all down in the medulla, okay? In the medulla. There's a lot of exchange going on here. So again, these descending and ascending limbs are all connected. They're all in this tightly woven space. There's a term called the countercurrent multiplier. Countercurrent means that there's fluid moving in opposite directions. In other words, water is moving from one into the other, and there could be movement of ions from one place to the other. Multiplication, because we're going to see that the deeper you go down in the medulla, the greater that pressure. And that's going to, quote, multiply this effect. What this is going to do is enable your urine to get concentrated. As the filtrate goes down the loop of Henle, it is going to it's going to go deeper down into the loop of Henle. And as it goes deeper down, the medulla gets saltier and saltier and saltier. I think of the medulla as a salty marsh. And these tubes are going down through this salty marsh. Solutes suck. So what's happening? As these tubes go down into this salty medulla, where's water going? Water is going to be pulled out, right? Solutes suck. So what's happening to the urine? Well, to the filtrate. It's not urine yet. What's happening to the filtrate? If I'm filtrate, I'm up here in the proximal tubule, and I'm going down the loop of Henle, and the deeper I go, the saltier the environment gets. Solutes suck. So what's happening to the water? Being sucked out into the medulla, isn't it? What's happening to my filtrate? It's becoming more concentrated. Okay, fair enough? So as we go down the loop of Henle, we're going to become more concentrated. So that's what this is going to do. This countercurrent multiplication idea is going to enable a highly concentrated urine to be formed. Because, as I've alluded to, we certainly can't lose those hundreds and 150 liters of fluid every day. Right? We're only going to urinate 1.5, 3 liters of urine a day. Do you remember back to day one? We talked about obligatory water loss. Maybe. We talked about where fluid compartments were. And the kidney must release, on average, half a liter of water a day. Remember we talked about metabolic water? That in the process of metabolism, you are forming water. If you look at the equation for cellular respiration, right? Glucose and oxygen make what? CO2 and water and energy. So we're making water. There's this metabolic water that we're forming. So we've got to get rid of this water. We've got to get rid of these waste products. So let's figure out, get a visual of this countercurrent multiplication. Now, we're in the nephron loop. Thick ascending limb. So let me go back. There's a thin descending limb. When you look at cartoons, some of them will make the nephron loop get thicker and thinner, okay, in certain places. So they're talking about a thin descending limb, a thick ascending limb as you come back up. So that's where we are. Now look what happens. The thick ascending limb actively transports sodium and chloride out of the fluid. Okay? Now it's interesting, the thick ascending limb is impermeable to water. So the sodium and chloride are leaving, but what can't leave? Water, right? It can't move. There's an impermeability to this region of the nephron loop to water. Now this seems counterintuitive at first, right? This whole countercurrent thing seems counterintuitive because what I just tell you is happening to our concentration as we go down the loop of Henle. The urine's becoming more concentrated. And now I'm telling you that as I come up, I'm getting rid of sodium and chloride, and I can't get rid of any water. So what's happening now? What's happening to the urine as it comes back up, the filtrate as it comes back up? The concentration becomes less. It seems really weird. Why would I first concentrate the urine simply to turn around and then make it less concentrated? But we'll see how all this puts together. What's happening to the fluid out here in the medulla? If I'm getting sodium chloride out, what am I doing? What's happening to the medulla? The environment's becoming more and more salty. Okay? So this sodium and chloride that's leaving the ascending tube is actually increasing that saltiness in the medulla. Okay, we'll put all this together. So here we are at a loop of Henle. We're going down, and see how it's thin, right? It's a thin descending limb. And look at the blue arrows. The blue arrows represent water reabsorption. Look what's happening to those blue arrows. Water is doing what? Leaving, right? Leaving the tube as it goes down. What's happening to the concentration of the solute? It's increasing, right? We're becoming more concentrated as it goes down. Okay. Then there is a ascending thick limb. See how it gets thicker in the cartoon? And look what the arrows tell us. Here, the orange arrows out are what? Those orange arrows are the solute being reabsorbed, not water. There's a selectivity to the movement of these molecules. On the way down, water can get out. On the way up, only sodium and chloride can get out. Again, the problem with this is that we have to imagine what is sitting right here. Capillaries, right? And what else is sitting out right here? What else is sitting out here? Kind of gray, isn't it? What else is sitting right here? More loops of Henle, right? So this is not happening in isolation. You've got movement of water from capillaries to tubes. All this movement's happening in this tightly packed area. So as we go down, water comes out. As we come back up, solute molecules can come out. So the deeper we go, the more concentrated we get. And as we come back up, the urine's actually becoming less concentrated. How is this happening? Well, here we are in the lumen of the loop of Henle, the nephron loop, and you can see that in this side, this is only on this side, right here, that sodium and chloride are being sent out, right? But water is not. Water cannot move across this portion. Okay, let's put this whole thing together. The descending limb, come back to it, is permeable to water and impermeable to solutes. So I just said that. As you go down, right, water can come out, sodium chloride can't. As you go back up, water can't move, but sodium and chloride can move. The water is moving by osmosis. And again, as we go down the tube, we're becoming more concentrated. As we come back up, we're becoming less concentrated. Plasma, the concentration of plasma is this number 300. I want you to see this number 300. Okay, 300. Referring to milliosmoles, we'll talk about the unit later. But 300. And as we go down the tube, as we go down the loop of Henle, look at the numbers. 600, 900, 1200. That fluid is becoming more concentrated. The higher the number, the greater the concentration of the solutes mixed in that filtrate. So as we go down, we're becoming more concentrated. What was happening here again? Remind me. What was happening down the descending limb? Water, right? See the water coming out? And then as we go back up, we get to the thick limb, the thicker area, and what could come out here? Sodium and chloride. Where is that sodium and chloride going? Out into the marsh, right? Out into the salty marsh, out into the medulla. Where is this water going? Where is this water going? What's sitting right next to it that we don't see? Vasa recta, right? Some capillaries. So that water is being sucked up. The sodium chloride is continuing to concentrate and making the medulla very, very salty. So as you go down, and as you come up into the distal convoluted tubule and in the collecting duct, we've actually made the urine less concentrated. Where does the urine have to go now? Where does the filtrate have to go now? Now that we're through the loop of Henle, where does it have to go? Through the distal tubule and then out the what? Out the collecting duct? The distal tubule and the collecting duct has the ability of increasing the concentration. And it's going to do this under hormonal control. You know about ADH. What does ADH do? ADH, antidiuretic hormone. Who makes, what organ makes ADH? I've already said it. Somebody asked about it earlier. ADH made by the hypothalamus, released by the posterior pituitary. And what does ADH do? Water retention. Made by the hypothalamus, right? And why is it going to be released? This is back to lecture 1. Remember we talked about water and balance and sodium retention and all that? Remember there are osmoreceptors. Remember this conversation? Day 1. Osmoreceptors in the CSF and in the blood that are going to be looking for concentration of our blood. And if the concentration of our blood is coming too high, what does that mean about us? If our blood osmolarity, if our concentration of blood solutes is getting too high, what do we not have enough of? Water, right? We're dehydrated. And so when our osmolarity increases in our blood, ADH is made. And what's it going to do? Tell our body to retain water to help restore that osmolarity. So that concentration of urine, although we did do some concentration down through the loop of Henle, we're also going to be concentrating the urine as it leaves through the distal tubules and through the collecting ducts. And this will be largely in the job of ADH. So ADH, we'll talk about ADH. We've heard about that before. ADH will be a big part of this. So by the time the urine leaves our body, leaves the nephron, we're sitting at about 450 milliosmoles. And our plasma was 300. So we've definitely concentrated the urine by the time it leaves the body. So let's take a look at this figure and try to put all this together. Just look at the numbers. 300. Right? That's like the same as our plasma. As it goes down the loop of Henle, water's leaving. Look at the little dots. The stuff in our urine is becoming more concentrated. It goes down as high as 1200 at the depths of the medulla. What is true out here? This renal medulla is very, very salty. What's that saltiness doing? Helping to pull the water out. And then as we're going up, how do we restore that saltiness? Remember, as we go up, what was coming out? Remember, we had sodium and chloride coming out as we went back up. Not water, but sodium and chloride. So that means that by the time we got back up here, we're actually 300. We actually have less concentrated urine. It makes really no sense. Like I said, the countercurrent thing is kind of counterintuitive. And then, as we leave the distal convoluted tubule and go into the collecting duct, oh, look what happens now. Water can leave again. So as we go down through the collecting duct, we're traveling through that salty marsh, and look what's happening to the solutes. Can you see how they're getting more concentrated in that cartoon? So we're getting more and more of these waste products now concentrated, and that's what's going to be leaving in our urine. I think that is tough to imagine. You got to look at it and put some numbers to it and just imagine what's happening to the urine. And specifically, what I want you to focus on is which part of the nephron's doing what. So when I say proximal tubule, you say reabsorption. When I say loop of Henle, it's concentrating it and going down that deep area. On the way down, what could come out? Water. What could not come out? Sodium and chloride. On the way back up, what could get out? Sodium and chloride. What could not get out? Water. As we come up to the distal convoluted tubule, we've kind of lessened our concentration. But then as we travel back down the collecting duct, we're going back down the middle of the medulla, aren't we? And as we go down that medullary region, it's very salty in the area, and more water is drawn out, leading to a concentration of our urine far greater than what it was when it came in. Now, why do we need that loop of Henle then? Is it important that we go from concentrated to more concentrated to less concentrated in the loop of Henle? Why do we need that loop of Henle? Let's put a little idea in our head. Let's try to make sense of this. Why do we need the loops of Henle? What were they doing? Helping to create a what? Salty medulla. And then what is it that's going right through the center of that salty medulla? The collecting duct. Right? And that's what's pulling that water out at the last minute so that our urine becomes more concentrated. This is, again, when you get to this part of the book, I definitely recommend that you check out these 3 or 4 pages on the countercurrent and the fluid and solute concentrations as you go through this part of the book. Now, back to the regulation of this movement, the regulation of the water. What is controlling how concentrated your urine is? We did this last week in lab. What happened to the folks who drank salt water? What happened to your output? Who in here drank water? Salt water, anybody? Talk to me, what happened? In general terms, not specifics, but just in general ways, what happened? Did you have to go to the restroom as much?
No, the first time I had an excessive amount.
Okay.
The second time I had a little, and then the third time I didn't have any.
Okay, so what's happening? She drank salt. It's like eating a bag of potato chips, right? Or any kind of salty meal. What's your body going to do with that increased solute? Retain water. Don't make this harder than it needs to be. So she drank salt water, right? And as a result, urine output went down, and your urine would have become more concentrated, and your specific gravity would have gone up. Who drank bicarb? Anybody? Anybody here drink bicarb? What happened, Caitlin?
I don't remember.
It should have also made it more difficult to urinate over time because what are you taking into your body?
Yeah, I should
Right? Solute. Right. So increased solute, right? Again, solutes suck. So salt or bicarb, what happened? Your body had to adjust to those increased solutes by water was retained. What happened with bicarb, though? What happened to your blood pH? Did it go up?
You couldn't go the last.
You couldn't go, so we can't read, you can't read negatives, right? Okay.
Oh, the two.
Yeah, so, okay, so probably with salt, no difference in pH, but what is bicarb? Back to lab 2 of 105. Remember the Alka-Seltzer Tums and Rolaids, and we are neutralizing stomach acid. If you're taking in a big old beaker of carbonated water, bicarb, you're basically taking in a lot of antacid. So what would happen to the blood's pH in response to that large amount of bicarbonate? Go up, right? Less acidic. So you should have, if you'd had enough urine to measure it and see the pattern, that's really why you folks did that, right? You did that just to see that result. Now, the people who drank more water, what happened? Lots of water, what happened? About 30 minutes later, more pee, and at 60 minutes, probably the floodgate opened, right? Lots of water. What happened to the amount of urine? Went up. What happened to the specific gravity? It went down. Okay. It's not meant to be too complicated. Okay. But it's pretty cool, the number of things that one can see in their urine and the number of conditions it can tell you about, right? I mean, liver health, kidney health, overall infection, all that stuff can be indicated by markers within your urine. So our body is definitely maintaining the amount of water reabsorption, which is affecting our volume of urine and the osmotic, right? How much solute there is in your urine. When I say more solute, you're thinking higher what? What are you measuring in lab? Specific gravity, right? Specific gravity is simply a measure of how much stuff, how much solute is mixed into your urine. Now, I mentioned to you that there's an obligatory water reabsorption. Where does most of your water get reabsorbed? Where's most of the water being reabsorbed? Within the nephron. Most of it, 65% of the water is being reabsorbed right at the very beginning in the proximal tubule. Along the loop of Henle, we saw some water being pulled out. Along the distal convoluted tubule, there's also water being pulled out. And there's even water being pulled out in the distal, in the collecting duct. So obligatory, 85% of the water that goes into your glomerulus and is filtered across into the proximal convoluted tubule, 85% of that by obligation, right, to sustain life is going to be reabsorbed. Now, if you're well hydrated, what's going to happen to your output? You're going to have increased output, right? If you're dehydrated, though, what's your body going to do? Try to recapture, hold on to that water. So obligatory water reabsorption simply says, boy, if we urinate 100 plus liters of urine a day, we're dead, right? We've only got about 3 liters of plasma. So what is the difference here? Where are the hormone signals coming in to regulate our water output? Again, most of this, the facultative. Who's had microbe? Anybody's had micro? What's a facultative anaerobe? Facultative organism is one that can kind of hang out in both situations, make choices, if you will, right? Facultative can make a choice, right? Facultative organism can choose to live in oxygen or not live in oxygen. So what does facultative water reabsorption tell us? You can make a choice. Right? So there's 15% of your fluid that either can be passed into your urine or that can be reabsorbed. And this control is going to happen in the distal convoluted tubule and in the collecting ducts. The hormonal control that your body has to retain fluid happens here. This is where the precise control comes, and it's going to absorb or adjust your urine output. So again, putting into pictures, everything in the proximal convoluted tubule and going down the descending side of the loop of Henle is the obligatory water reabsorption. About 85% of your water is going to be reabsorbed by the time it hits the bottom of the loop of Henle. Remember, coming up, you can't reabsorb water, right? It's impermeable to water on the way up. And then over here in the distal convoluted tubule and down the collecting duct is where, under hormonal control, the body can retain more fluid. If you're dehydrated, what's going to happen? Is there going to be more of those blue arrows reabsorbing water or fewer? If you're dehydrated, more, right? Your body's going to retain that fluid. If you're well hydrated, let it go, right? No need to reabsorb anymore. We've got the 85% that we need, and now the rest of it can just simply go into your urine. What hormone is controlling this? I've already mentioned one, right? And that is ADH. Now, no water is reabsorbed in the DCT or in the collecting duct without this. Okay, you need ADH to do this. If you don't have ADH, what would happen? If your hypothalamus did not make ADH, what would happen? What would you be doing all the time? Peeing. There's a form of diabetes, not related to sugar metabolism, but related to over excessive urination. Have you ever heard of diabetes insipidus? It's a different form of diabetes, not related to sugar, but related to osmotic regulation, and these folks don't have any ADH, right? Their hypothalamus doesn't make ADH. So what happens? They have no way of controlling this flow, so what's always happening? They're urinating constantly, liters and liters and liters a day. They're always drinking, and they're always urinating. They don't have control over this process.
What do they do for people like that?
Well, they just have to keep them hydrated. I think there's some, there might be some medical things they can do now to help minimize the problem, but they're just peeing all the time. I mean, up to 20 and 40 liters a day. Yeah, they're always taking it in though. They're having to take it in, right? Their body is driving them to drink, and they're constantly urinating. Take a look at this image. What's happening in this picture? We've already said this, right? We got to get this in our head. What's happening? Look at the water. Water's coming out from this point. What do we call this? All the water movement out on that side was called the what? Obligatory water reabsorption, 85%. And then what's happening along the rest of the way? The facultative, right? The choice, if you will, what the body's choosing to do. But look at the little dots. The dots are telling you what? The more dots you see, the more concentrated the filtrate is. So again, it got very concentrated down here in the bottom of the loop of Henle. It becomes less concentrated on the way up. But then what happens is it comes back down. What if you didn't have reabsorption of water? What would happen? Your urine would become, that's what this is showing. This is no ADH. Look at can you see behind all my scribble? Look at the solute concentration. It's very low, isn't it? You're basically peeing out water. You're not concentrating the urine. You have no control over this. So you'd have very, very large volumes of very dilute urine. Another way of saying it is that the specific gravity would be very low. Almost what? What number? What's the specific gravity of pure water? 1.000, right? So basically this urine is almost pure water. It's not being concentrated, is it? So with ADH, right, what happens is how does the distal convoluted tubule and how does the collecting duct get more water? There's actually a molecule, a protein called aquaporin. Well, what would an aquaporin be? IN, it's a protein, and it creates pores for water. It's basically a channel for water to increase. Now we know from the first couple lectures of AMP1 that water is able to move across the membrane pretty easily, isn't it? Water can move across the plasma membrane even though it shouldn't, right? Even though it's a polar molecule, even though it doesn't make any sense, water does move across the membrane. But what ADH does is it inserts these aquaporins into the tubule, and that increases the water reabsorption. And as a result, we reabsorb more water, our urine is more concentrated, right? And we are able to maintain also, we're also able to maintain our salty medulla. Because what would happen if we couldn't maintain that osmotic concentration then as the fluid moved down through the tube, we wouldn't get it concentrated. So this is what happens with ADH, right? And I don't want you to think that ADH is only released when you're, well, like in the desert, dehydrated, right? There's a homeostatic control happening here. So look now at the solute concentration. The number was about 300 up here. As it goes down, right, more and more concentrated. As it comes back up, it actually becomes less concentrated. We talked about this. Right about here in the distal convoluted tubule and in the collecting duct, that's where ADH can work. And everywhere where you see these blue arrows, it is water reabsorption, but it's being increased by the presence of ADH, because what is being put into the membrane? These aquaporins. These aquaporins allow more water to come out of the collecting duct. As a result, the urine becomes more and more concentrated. So what we put out now is much smaller volumes, more concentrated, higher specific gravity urine. Normal urine, 1,200 mils a day. Okay. Again, these are that normal physiological 25-year-old, 150-pound male, but about 1,200 mils a day, 1.2 liters. It can certainly be more if you're drinking through more water throughout the day. It can be less if you're not in the habit of drinking water. It's been said that if you are thirsty, you're already dehydrated, right? That you should be kind of pushing a little bit of water all the time, even when you're not thirsty. So people who walk around with a water bottle and are kind of, just sort of by habit sipping on it are probably doing very well for their kidneys. And those who wait till they're thirsty and their mouth gets dry have probably waited, and their osmoreceptors are already targeting or triggering that thirst response. And that thirst response is only triggered when you have already been, quote, dehydrated. So if you're getting thirsty, you're already dehydrated. You're already making ADH. Your body's already putting these aquaporins into the membrane. So if you're exercising, if you're drinking more, of course, these numbers are going to fluctuate quite a bit. Look at the specific gravity. We saw these numbers upstairs. pH, average of 6, right? Somewhere around 6, definitely more acidic than your blood. Can it go alkaline? Can the pH of your blood go alkaline? Yeah. What did that tell us? Remember in the lab, there were very acidic, let me quiz you on that. Very acidic urine below 3 suggested what? Remember on the chem strip, there was the what ifs? And if the pH was really low, like 3 or lower, one of the suggestions was maybe acidosis, right? And that makes sense. A diabetic may have more ketones, might be acidic. A person with emphysema, right, might have more acidic urine. Why would that be? My RT people. They're holding on to CO2, right? And the more CO2 they hold on to, they're not getting out that CO2. It's becoming more acidic. The blood's becoming acidic as a result. Specific gravity. Well, what did a high pH mean? Over 7? What did it tell you in the lab manual? Alkalosis, but it would be because of an infection, right? Wasn't there a urinary tract infection or urogenital infection, I think was one of the things listed on there. Specific gravity, your urine really would rarely be 1.000, right? There's always something dissolved in your urine. So 1.003 up to 1.030, that's going to be considered rather normal. Water content, 93 to 97%. Volume, 1.2 average per day, 700 to 2,000 or so being the average, at least 500. Right, back to lecture 1, day 1, the obligatory water, about 500 milliliters of water your body has to get rid of every day. That's why you can die of thirst much faster than you can die of hunger, right? It doesn't take very long to die. If somebody's going to go on a hunger strike, let them go. It's going to take weeks. Urine, sterile, kind of. Why would urine not be sterile? If one had a urinary tract infection, right? There's already infection along the bladder, along the ureter or the urethra, then there would not be sterile urine. But for the most part, when your blood is sterile, right? Should be. So if your blood is sterile, therefore, so should your urine as it is filtered out the glomerulus. So unless there's bacteria along the urinary tract, urine should be essentially sterile, lacking of bacteria. So let's put it all together. Last 5 minutes. Let's put it all together. What are the processes? What's the first thing that happens in the kidney? Blood comes in. and is filtered at the glomerulus. The number I want you to know here is 300. That 300 milliosmoles was what we keep seeing in all the figures. Your plasma is about 300, your filtrate's 300, right? What comes across. Remember, the plasma and the filtrate are essentially the same thing, aren't they? Would you agree that in the Bowman's capsule, the Bowman's capsule is essentially a pretty good representation of the plasma, except for blood cells and proteins. But the electrolytes would be very, very similar. Then we're going to go down the proximal tubule. As we've said, what's happening there primarily? Reabsorption. Water's moving by osmosis. We've got the movement of other electrolytes, small proteins. Then we're going down the nephron loop. Again, we're going to have that obligatory water reabsorption, concentrating the fluid as it goes down. I like this image because at least now it shows me vasa recta, right? And we're remembering that as down here in the medulla, we still have this vasa recta. Now, look at the blood flow here. Which way is the current of the solute moving? Right, down the tube, right? And look what's happening here in this vasa recta. The blood flow is kind of moving in the opposite direction. So there's this counter movement of molecules. But regardless, look, where's this water going? As water comes out of the collecting duct and out of the distal convoluted tubule, where's it going, guys? Right into the vasa recta. Can you picture it? Right, the vasa recta, that series of capillaries that's wrapped around the loop of Henle. Two kinds of nephrons. Which nephrons are involved with maintaining the salty medulla? What kind of remember there's two kinds of nephrons, two flavors. Yeah, juxtamedullary, because what do the juxtamedullaries have? The deep loop of Henle and the vasa recta. So the saltiness of the medulla is maintained by the 15% of your nephrons that are juxtamedullary. Whereas your your cortical nephrons are pretty much all about reabsorption and secretion, aren't they?
Does the concentration get is it as thick in the cortical one as it is in the?
No, so you're saying the urine? You're picturing with me, everyone's picturing with me a very wimpy loop of Henle, right, in a cortical nephron. Because it goes to go down as deep, it doesn't get as salty. Good. So it has to go into the deep end of the pool, right, to get high pressure. So if it doesn't go down very deep, there isn't as much of a concentration. It's really those vasa recta and those deep loops of Henle that are maintaining that saltiness of the medulla. So then we're going to continue. What do we have? Then we're going up the ascending side, as we already said. This is just a review. We're going to be actively transporting. That means energy is required, a lot of mitochondria. Actively transporting sodium and chloride. We make adjustments throughout the distal tubule and the collecting ducts through ADH. And we just saw the vasa recta, how it's helping to do the exchange of that really salty medulla. So again, here we see this movement of molecules, the water, back into the vasa recta. And again, at least here, this image is giving us the idea that the vasa recta is in direct contact with both a ascending limb and the collecting duct. So I've never seen a good cartoon representation of the kidney that helps to bring this to appreciate how tightly packed these vessels are side by side. This one is a little better. Okay, again, we're seeing the complexity. You've seen all this before. But again, it's just telling you where is this stuff going. So water coming out is going into the vasa recta. Solutes coming out on the ascending side are also going into the vasa recta, right, or into the tubular capillary. So this stuff all has a place to go. It's all exchanging here within the medulla. What can go wrong? Got just a minute. Renal failure. What's not happening? Kidneys are shutting down. What are you not doing? You're not urinating. You're not filtering. Your acids are rising, right? Those metabolic acids are not being gotten rid of. If they're not getting rid of those metabolic acids, your blood's becoming more acidic. You're becoming very acidotic. At what point are you dead? 7.4 is homeostatic normal for blood pH. How low can it go? about 6.9, okay? And it can go up about as high as 7.9. So there's about a plus or minus half a pH unit for life, right? Trying to keep it at 7.4. If your kidneys aren't working, what else are you becoming? What hormones are you not making? Renin. Therefore, your blood pressure has some issues. If you're not making renin, what's going to happen to your blood pressure? Renin's pushing it up, wasn't it? So without renin, your blood pressure is going to drop. That's making the kidneys even more pissed off, right? Because they're not getting the perfusion, they're not getting the volume that they need. You're also not making erythropoietin. So what's that affecting? Your red blood cell, and you're now anemic, right? Because you're not keeping up with your red blood cell production. So we all see those connections, right? Anemia, lower blood pressure. Now, there would be hypertension, though, in this situation. So I just misspoke, didn't I? So without that renin, there would be, though, some hypertension occurring because the blood flow is not going to the kidneys as well. And with that pH, what's going to happen? What's the problem with that pH? The central nervous system starts to be affected by that acidotic environment, causing seizures and coma. So we'll pick up with a couple of things there. What I want to do on Thursday is review this quickly, get us back into thinking about the kidney, answer any questions. Now, I know there'll still be a subset of you on Thursday that are dealing with the lab exam on Thursday night. I know that. But hopefully, you'll be able to think through this a little bit. We'll go through a few more of these ideas about dialysis and about when the kidneys are failing, and that'll bring us into understanding the acid-base regulation that we'll be finishing up with. So I'll finish up with this, review the kidneys, and then move into the last little bit of chapter 25 on acid-base regulation. That'll be our last content for this semester.